Hello student, welcome to this lesson. We want to look at the structure and function of the blood vessels. First, we have various types of vessels like arteries, capillaries, veins. Those are the major. We shall begin by looking at the arteries. The arteries are blood vessels originating from the heart and usually carry blood away from the heart. They are deep seated in the muscles, that is, they are deep inside the tissues. The aorta is the main artery and carries oxygenated blood from the heart to all parts of the body. Arteries carry oxygenated blood except the pulmonary artery, which carries deoxygenated blood to the lungs. That means the blood that has been brought from all parts of the body to the heart is pumped out or to the lungs to receive oxygen through the pulmonary artery. Arteries have a thick muscular wall which has elastic and collagen fibers that resist the pressure of the blood flowing in them. The pressure is due to high or due to pumping action of the heart. The pulse, or the number of times the heart beats per minute, can be detected by applying pressure on an artery next to the bone, for example, by placing a thumb on the wrist of your hand. Artery has 3 layers. The innermost layer of the artery is called endothelium and is smooth. It provides a smooth lining which offers the least possible resistance to blood flow. And this layer is found in almost all the blood vessels, that is endothelium. This is the structure of the artery, that is the endothelium. We are talking about the innermost. The middle layer is composed of elastic fibers and smooth muscles. The outer layer contains collagen and inelastic tissues or fibers. So when ventricles contract, the muscular layer of the arteries relax, stretching outwards to allow blood into arteries. So that is when the ventricles, especially the left ventricle, is all the ventricles contracting so that blood can flow out. That is when the arteries relax, then they expand to allow in blood. And when ventricles relax, the muscular layer of the arteries contract, pressing inwards, and thus the blood flow, forcing the blood flow forward, producing a pulsating action in the arteries. The muscular walls of arteries is under the influence or the control of hormones and nerves to alter or change the diameter of the arteries. Hence, blood flow to the body organs is regulated. We've said aorta is the main artery and forms a branch that supplies blood to major body organs. These branches from the main aorta or from the aorta divide to form arterioles. The arterioles branch further to form capillaries that ramify the body tissues. So the capillaries are numerous or many and very close to the tissue, and they facilitate exchange of substances between the tissues and the blood, because the capillary wall has this endothelium, which is one cell thick or very thin. You can see there is exchange of materials. Oxygen from the red blood cells is diffusing into the cells, and the cells has more concentration of carbon dioxide and waste. Therefore, they have to diffuse into the capillary for transportation back to the heart. So this thin layer of endothelium, which is one cell thick, provides a shorter distance for exchange of materials. The capillary walls are also made permeable to water and molecules of small size in solution. That means it only allows substances that are of small molecular size, which are in solution, but not large ones. And also water can pass through the capillary wall. Branching of arterioles into capillaries increases the surface area of the capillaries and the blood pressure in them. Therefore, they have a narrow lumen, and that means blood flow in capillaries is under high pressure due to this narrow lumen caused by branching of arterioles into capillaries. This high blood pressure forces water and dissolved food substances out of the blood to form tissue through a process called ultrafiltration or pressure filtration. Part of the tissue fluid passes back into the capillaries at the venule end. The tissue fluid is the watery liquid similar to plasma in composition, but has no proteins, red blood cells, and some white blood cells. The excess tissue fluid drains into small channels called the lymph capillaries, which empty their contents into the lymphatic vessels. Capillaries then join to form larger vessels called the venules. These venules join to form veins that transport blood back to the heart. The phagocytic white blood cells are able to squeeze in between cells of the capillary wall into the tissue fluid. The tissue fluid bathes cells of tissues and forms medium of exchange of materials between blood and the cells. The cells obtain oxygen, glucose, hormones, amino acids, vitamins, and mineral salts from the tissue fluid. Cells also release carbon dioxide and urea into the tissue fluid. Ultrafiltration results in a drop in pressure such that at the venule end of the capillary bed, the blood pressure is lower than that of the tissue fluid. Proteins retained in the capillaries during formation of tissue fluid exert a higher osmotic pressure at the venule end of the capillary, causing much of the tissue fluid to be drawn back by osmosis. That means carbon dioxide diffuses across the capillary walls into the blood. The remaining tissue fluid drains into the lymph vessels and joins the lymphatic system to form the lymph. Venules from the capillary bed join up to form the veins. So we move to the veins, and veins carry deoxygenated blood from the tissues to the heart. So that means this blood is lacking oxygen. So veins carry deoxygenated blood from the tissues to the heart, except pulmonary vein that carries oxygenated blood from the lungs back to the heart. And they lie close to the body surface. This is the structure of a vein. It has the smooth muscle on the middle, endothelial cells on the inner, and collagen fibers on the outer part. You can see the lumen is narrow, actually wide, but that one over artery, or actually the lumen of a vein is wider, and that of a artery is narrow. Veins have a wider lumen than arteries. This offers minimum resistance to blood flow. Their walls are thin than those of arteries. That means blood pressure is low. Forward flow of blood in veins is assisted by contraction of skeletal muscles. Hence, it is important to exercise. Veins have valves along their length to prevent backflow of blood. That is, to ensure blood flows towards the heart. Generally, blood in veins contains high levels of metabolic wastes and low levels of nutrients. Exceptions to this are the renal vein and the hepatic portal vein. So renal vein is carrying blood to the kidney. Or actually, renal vein is having a low level of nitrogenous waste, as this will have been removed in the kidney. Hepatic portal vein carries blood rich in nutrients that have been absorbed in the gut or the digestive system. Let us look at the structural differences between the artery and the vein. Arteries have thick muscular walls with more elastic fibers. On the other hand, the walls of veins are thin, less muscular, and less elastic fibers. The walls of the arteries have no valves except at the base of large arteries leaving the heart. But the veins have valves at intervals throughout their length. Arteries have a narrow lumen, whereas veins have a wider lumen. Then, functional difference between arteries and veins. The artery carry blood away from the heart. Vein carry blood towards the heart. Artery carry oxygenated blood, except pulmonary artery that carries blood to the lungs. Veins normally carry deoxygenated blood, except the pulmonary vein that carries blood from the lungs to the heart. Blood flows rapidly under high pressure in artery, but in veins, blood flows slowly under low pressure. In artery, blood flows in pulses, whereas in veins, blood flows smoothly with no pulse. Artery have the ability to alter the lumen, whereas vein have no ability to alter or to change their lumen. So we have a practical activity you can be able to carry, that is displaying the circulatory system of mammal, where we need a freshly killed animal like rabbit, and then a dissecting board, pins, all these apparatus. Then the procedure is to pin the animal so that the ventral side is up on the dissecting board. And then the animal is cut along to open up the abdominal cavity and display the internal organs. Then cut and remove the diaphragm, taking care not to damage the heart and the blood vessels. You have to identify organs like the heart, liver, spleen, lungs, and the kidneys. Then identify the main blood vessels like the pulmonary vein, coronary artery, renal veins, hepatic portal vein, pulmonary artery, hepatic artery, the mesenteric vessel, aorta, and vena cava. Then identify and name the blood vessels carrying blood to the following organs: the brain, forelimbs, lungs, kidney, the liver, and the hind limbs. And then examine the external structure of the heart, paying attention to shape and thickness of its walls, the aorta, or the auricles or the atria, as well as the ventricles. You have to identify them. Identify the coronary blood vessels on its upper side. And then after having seen all those external parts, you have to dissect the heart to expose the internal structure of the heart. Then cut through section of the pulmonary artery down into the auricle, then into the right auricle or right ventricle. Cut through the aorta down into the left auricle and then into the right ventricle. Then examine the exposed internal structure of the heart and identify this part, the chambers of the heart vessels, or actually valves, bicuspid, tricuspid valves, semilunar valves, and the tendons. Then you have to draw and label internal and external structures of the heart and even show the direction of blood flow using arrows. So that marks the end of that interesting lesson of the blood vessels. Goodbye.